Hey everyone. So today we're going to be doing the exploring photosynthesis and plant pigments lab. So I'm going to go ahead and share my screen and we're going to get started with this. So you can find this lab in your week 9 folder on the e-learning page. Again, the title is Exploring Photosynthesis and Plant Pigments. And so let's get started. So the overview of this lab is really to begin to explore photosynthesis and pigmentation. So I'll let you read through the overview. But the outcomes for this experiment, one, we're going to determine the number of pigments in a sample using paper chromatography. And that's something that you guys might have done in high school. We're going to formulate a hypothesis predicting the effect of varying light conditions and dissolved carbon dioxide concentration on the rate of oxygen production as a proxy for the rate of photosynthesis. So pretty much we're going to be using different light conditions and different carbon dioxide concentration conditions to measure the rate of oxygen production and use that as a way of gauging the rate of photosynthesis. And then we're going to collect and graph our data. So pretty much the time requirements for this, we have about 20 minutes of prep, which I've already done. Activity 1 is going to take about 20 minutes. And activity 2 is going to take about 120 minutes. And so I'm going to lay out pretty much how I did all of this and try to help you guys shave off some time there. But personal protective equipment or PPE for this lab are going to be goggles, gloves, and aprons. The big thing that you need to be worried about is the 2-propanol or the isopropanol, 70%. You want to be careful with that. And using a pin, you just don't want to stab yourself, right? So the background here is going to go into things like chloroplast, photosynthesis, the light-dependent reactions of photosynthesis. So looking at pretty much the electron transport chain within chloroplasts. And then it's going to look at the Calvin cycle or the dark reactions. And then eventually a little bit on pigments and chromatography. Interestingly here, we're talking about wavelength and wavelength absorption. It's always interesting, when you see a green leaf, the leaf itself is not absorbing green light, right? Wavelengths within the green spectrum of light, right? That's what's reflected off. That's what we see. But what I'll do is I'll give you guys a couple minutes to read through the background yourselves. So you can go ahead and pause the video. And then when you're ready to go through the procedure, you can unpause and join me back here. All right, so if you're back with me, I assume that you read through the background and we're good to go. So materials wise, what we need for this, we need the lab kit materials for the Exploring Photosynthesis lab. From the equipment kit, you'll need two 250 milliliter beakers, your 25 milliliter graduated cylinder, your test tubes and your test tube rack, a ruler, and your grease pencil, and some other things that you need. Dish soap, tap water, a coin with a ridged edge, so something like a dime or a quarter, not a nickel or a penny. A light source, so you're going to need to set up a light environment. You're also going to need, it's not here, but you're also going to need a dark space. So what I did was I just cleared a little space in a kitchen cabinet, and that's going to serve as like my dark space because I can shut the door. And on my desk, I just have my desk lamp set up for my light space. You're going to need paper towels, pencils, plant leaves. So I just have like spinach leaves that you would get in like a salad kit from your food store, a timer or timing device, so your cell phone, scissors, and a small bowl filled with tap water. So a bowl this size is more than large enough. Pretty much, we're going to be punching some holes in our spinach leaves or our green leaves. And we're just going to keep the punched out leaf in the water to keep it hydrated. So safety, goggles, gloves, apron. Isopropyl alcohol is highly flammable. This is alcohol that you would normally keep in your house for like disinfectants and things like that. Don't get it in your eyes. Don't inhale it. Don't eat it or drink it. The normal things. All right. So activity 1, plant pigments. Now, I've already done a lot of the preparation work, and I'll go through that, like what I did when I get to it. A lot of the prep works with activity 2, and I'll kind of tell you like when you should be doing the prep work to save some time if you're just starting here and you haven't done any prep work. All right, so activity 1, plant pigments overview. In this activity, paper chromatography will be used to determine if a green leaf contains only one pigment or a mixture of pigments. Okay, so prep. Spinach leaves are recommended, but any kind of tender, dark green leaf will work. Refrigerate the leaves prior to use. Clean and sanitize your work area. Gather needed materials. And don't touch the chromatography paper and like rub your fingers in it. Because the oils can mess with the wicking of it. And a tip here, cut a piece of chromatography paper lengthwise into 2. So they say that they gave you 2 pieces, but I only have 1, but we're going to cut it lengthwise into 2. And the reason they say this is to you have a backup piece if something goes wrong. All right, let's cut lengthwise into 2. Okay, so procedure. At 1.5 centimeters from the bottom of the chromatography strip, draw a faint pencil line across the strip. All right, so things we need our pencil and we need our ruler. So we're going 1.5 centimeters. So we're on our metric side. Okay. So that's going to look something like that. That will be your origin line. You can click the video to pretty much watch what I did and I'm about to do. So you're going to lay a fresh spinach leaf on top of the origin strip. Yeah, so I'm just going to angle my laptop down here. Okay, so we can see it there. We're going to lay the spinach leaf over where the origin strip is, and we're just going to roll the edge. Not hard, just back and forth. And we should get a nice transfer of pigmentation. So let me show you guys what that looks like here. So you can see the green pigment from the spinach leaf, right? So it's not a lot of hard rolling. You're not rubbing the leaf into it. You're literally just taking the leaf or the coin and just rolling it. Okay. They say press firmly. You really don't even need to press that firmly. If the line is not present or faint, the leaf you're using might be like really dehydrated and not good to use, or maybe you're just not pressing hard enough. Okay, so we're going to place the test tube in the test tube rack. So I have the one that I'm going to be using up front. Add about 1 centimeter of isopropyl alcohol, approximately 20 to 25 drops to the plastic test tube. The isopropyl alcohol will serve as the solvent that wicks and separates the plant pigment from the leaf of the paper. All right, so we're going to be working with isopropyl alcohol now. So I'm going to make sure that we put on our gloves. You can also really make sure that you put on your apron and goggles here. Okay. The other thing is, it seems like they pack the isopropyl alcohol with the pins. So just be careful when you're removing this from the bag. You don't want to stab yourself. They tape the pin to the isopropyl alcohol. I don't know if they did that for everyone, but okay. So I'll leave the pin there. And we're doing 20 to 25 drops. You have to really squeeze to get drops out of this, so I don't think you need to worry about overfilling. Okay. My 25 drops. Put that on my paper towel. All right, see, you got about 1 centimeter in there. All right, if you really want to verify, you can use your ruler. And that's about 1 centimeter. So that was 25 drops, and that was about 1 centimeter for me. Okay, place the strip on a paper towel and mark the solvent front with a pencil before this. Sorry, wrong spot. Okay. 7. Insert a straight pin into the center of each chromatography strip around 2 centimeters from the top. All right, well, I only have 1 pin. So I'm going about 2 centimeters from the top. Doesn't help that I can't grip the ruler now that I'm wearing gloves. Okay. I'm gonna get a pin out of here, hopefully. Okay. Pin. We're going about 2 centimeters and in the middle. Okay. Careful not to rip your paper and or stab yourself here. Okay, so you'll have something like that. Carefully lower the chromatography paper strip with the line oriented to the bottom, so this side down, which makes sense. down the center of the test tube and into the solvent. So the strip should be in the center of the tube, not touching the walls. Adjust the strip's position by moving the pin side to side. The origin line with the pigments from the spinach must be above the liquid level, yet the bottom of the paper must be in the liquid. So we're going to allow the solvent front to wick up the chromatography paper through the vivid green line until the solvent reaches within a centimeter from the top. So once it's past the pin is what we're going for, right? This should take around 10 to 15 minutes. All right, so what we're going to do is we're going to take this. We're going to drop this in. Okay. Apparently, I need more isopropyl alcohol in there. So that's good. How am I going to do this? All right, so we need more than 25. It's really hard to get it to not be on the sides, but so after some finagling, we're in there. Okay, so we're wicking. We got something like that. So all in all, it took about 30, like 5-ish drops, 30, depending on, I guess, how good your drops are. But just be wary of that. So we're going to let that sit for about 10 to 15 minutes. All right. So while that's sitting, we're just going to put that over to the side, keep an eye on it. I suggest we move on to activity 2, right? Because there's no point in sitting there waiting for 10 to 15 minutes for no reason. So activity 2, we're going to be doing the leaf disc assay. All right. And so in this activity, we're going to be conducting an investigation to examine the rate of photosynthesis in various light conditions and CO2 concentrations. Great. So there's a lot more prep to this, and I suggest this is like this window, you actually take the time to do some prep. All right. So again, we're going to get some more spinach leaves. We're going to clean and sanitize our work area. We're going to set up our light storage area. So create a storage place under a bright lamp. Use a grow light if you have one, if you're growing plants, or a desk lamp. And then we're going to be, you want to use around a 60-watt equivalent bulb. Whatever you have is fine. Whatever you have is fine. Don't go buying light bulbs for this. And then you're going to set up a dark storage area. So like I said, I made a little space in my cabinet, and that works fine. You could do like under a box in a dark room, whatever it might be. All right. So you're going to prepare soapy water by adding a drop of dish soap to about 20 milliliters of water in a medicine cup and mix the contents. So I have that here. And then you're going to be making your bicarbs, your 0.24% and your 0.12%. So steps 6 through 15, you're going to be prepping them. But at the end, you should have a 12 milliliter beaker, or a 0.12% beaker with 250 milliliters in it. And you should have a 0.24% with about 125 milliliters in it. And then you're going to prep and label your syringes. So your syringes, mine came in bags, and then the bottom caps came separately. I'm just going to take a peek at our wicking. Yeah, we're wicking very nicely. Okay. So light experiment, right? So if you do that and that thing's still going, move on. All right, let's look at this light experiment. So cut 40 to 50 leaf discs from the healthiest looking leaves. All right. So I've already done that as well. They give you plastic straws, but I used a metal straw that I just had at home. And that worked well. Plastic straw will also work just as well. You're going to fill a small container or a bowl with water. Lay the leaf on a hard surface, protected by a paper towel because you're going to be releasing pigment. Press the end of the straw. So you're literally going to be taking the leaf right on your hard surface, pressing down. Right, I could just do it on my thumb. And what you do is you'll punch out a little hole. Let's see if I can get this on camera here. A little punch out of the leaf. And you're going to be doing that 40 to 50 times, right around 40, but if you want a few extra just to be safe. The big thing here is, and once you punch them out, put them into the water container to sit. It's important that you're using the same size straw throughout. So that's the only thing. And if you need help with this procedure, if you need an additional video, you can watch a video here from Carolina. It's a good video. But all right, so we're going to move on to step 3. We're gently going to place 10 leaf discs into each of the 4 syringes. All right, so we're going to be using the straw to gently position the disc close to the syringe tip. So what we're going to do, we'll go with A first. Okay, we're going to take the plunger out. Okay, we're going to take our straw, and we want 10, right? We're just going to pick it up on the tip of our straw. Probably shouldn't have picked a straw that changes colors. And we're just going to drop them in the bottom of the syringe, right near the tip. If the thing gets stuck within the straw, you can just blow it out. It's 4. This might seem tedious, but it's not too challenging. Nine. Okay, we're just going to want to get these all to the bottom as much as possible. helps when they don't go in the straw. Okay. So that's 10 in the bottom here. Okay. We're all just chilling in the bottom. Okay. We're going to replace the plunger. Okay. Push towards the tip without smashing. So you just want to go as far down as you can without crushing them. All right. Fill syringes A and B with solution from the 0.24 beaker. All right. So with this setup, I'm just going to be showing you one, and then we'll chat about doing the other ones. We're going to check that, make sure we're still good. Okay, so we're going to fill with 0.24. So we got our syringe, we got our 0.24. Place the syringe tip in the liquid, pull back to overfill the syringe to the 6 milliliter mark. So we're going up to the 6 milliliter mark. Okay. Okay, we're up at the 6 milliliter mark. Tap out any bubbles. All right, so we don't have any bubbles. Push the syringe tip firmly into the syringe stand. So we want to get as much air out of this as we can. All right, so we're just going to eject up until, do this in view. We're just going to eject up like a doctor would in the office or a nurse would until we get a little bit of water out. Okay, now all the air's out, so then we're going to draw back to 6. Okay, if we see any bubbles, we'll tap that out. But you notice our leaf little cutouts are floating. Okay, trying to get as many bubbles out as we can. All right, so push the syringe firmly into the syringe tip. Pop it on. Just make sure that we are bubble free real quick because there was some bubbles in there. So again, tap up the bubbles, eject. Right. We have to got a very large bubble. Well, you get out all the bubbles you can. Okay. We're going to pop that in. Okay. We're going to put that off to the side. And then we're going to do B as well, same thing. And then for step 6, we're going to fill syringes C and D with the solution from the 0.12% beaker. But you're going to be doing the same thing. So go ahead and pause the video and join me back here when you have all 4 of your syringes with your leaf cutouts filled with the appropriate solutions. All right, great. So welcome back. So once you have all 4 done, we're going to move on to step 7. Okay. So for each syringe, pull back gently until a slight vacuum pressure is felt. Do not pull the plunger past 9 millimeter mark. So while still in the cap, we're going to pull back until we feel a nice vacuum pressure, which you can feel pretty quickly. This vacuum will pull any air out of the leaves, small air bubbles. And if you notice, when you do that, you're getting a ton of air bubbles in the leaves. Okay. So while you're doing this, you want to gently be tapping or shaking the syringe to work to get as many of the air bubbles out as possible. So slowly release the plunger, allowing the syringe to return to atmospheric pressure. So I would also suggest putting your finger over the cap. Again, you know, we're just pulling back, we're flicking, we're trying to get all the air out. Okay, and now finger over the cap, we're slowly just going to release the plunger. And if you notice, some of your discs start to fall because we're taking all the air out of them. All right, so we're going to remove the cap. We're going to push out now any air. So we're going to keep flicking all of our bubbles up to the top. We're going to push out any air. All right. Then we're going to recap it. We're going to do the same thing. Draw it back, create a vacuum. Okay, again, not going past the 9mm mark. Okay. While we're doing that, remember we're tapping. We want all the air to go up to the top. We're slowly going to release it back. Our discs will fall. Our air bubbles up. Eject air out. Redo it. I would suggest doing this 5 times. You want them to be as dehydrated as possible. Or sorry, you want to get the oxygen out of them as much as possible. Three times. Remember tapping or shaking. Slowly release back. And each time there should be less and less air within the vial itself. Within the syringe itself, excuse me. So that time there's barely any. We're gonna do a final time. Okay, barely any again. All right. After the disc like sunk, turn the syringe tip up on this. After the discs have sunk, turn the syringe tip and syringe stand up. Then remove the syringe stand. Remove any air bubbles and then fill the syringe to the 5 milliliter mark with the designated solution and replace to the syringe stand. Yep. So if you're over 5, eject out to 5. If you're under 5, refill to 5. Okay. So we're going to, I'm a little over, so I'm going to eject out to 5. And then replace the stand. All right. The important thing now is that you guys go ahead and you do that for syringes B, C, and D. Doing the same thing. And also remember, A and B are to 0.24%. C and D are to 0.12%. Right, so you're going to repeat steps 7 to 13 for each syringe. At this time, each syringe should contain 5 milliliters of solution, no air bubbles, and all the leaf discs should have sunk to the bottom of the syringe. So like that. Okay. Place the syringes at the same time with the syringe stand down. So this is for the light one. So syringes A and C. Place the syringes at the same time with the syringe stand down about 10 centimeters from the light source. This is for A through D. About 10 centimeters from the light source. At 5-minute intervals, tap the sides of the syringe and then count the number of leaf discs that are floating. All right, so you're going to set them down at your light source, about 10 centimeters from your light source, all next to each other. And every 5 minutes, you're just going to tap the side of the vial, the side of the syringe, and see how many discs are floating. And you're going to collect data every 5 minutes for 60 minutes or until all the discs are floating. And you're going to record that in data table number 2. Now you're going to move on to the dark experiment. So you're going to repeat the experiment above, except that instead of placing the syringes 10 centimeters from a light source, you're going to place them in a dark location as described in the prep section. And every 5 minutes, you're going to remove the syringes from the dark location, tap the sides of the syringe, and then count the number of leaf discs that are floating. And so you're going to collect data every 5 minutes for 60 minutes or until all the discs are floating. And you're going to record data in table 3. All right. So the final part of that is you're going to calculate the average number of discs floating per tube and graph the results to depict the number of floating leaf discs for each treatment over time. So pretty much what you're going to do in activity 2 is you're going to pretty much do everything up to step 13 twice for the 4 syringes. So you're going to be filling and pretty much pulling all the air out of each syringe or a syringe 8 times. All right, 4 for the light, 4 for the dark. And then just disposal and cleanup for when you're done this. So obviously I'm not going to let this video run for 2 hours. But if you have questions with that, feel free to reach out. Disposal and cleanup. So you're going to dispose of the buffer, baking soda solution, leftover isopropyl alcohol, down the sink with excess water, dispose of the chromatography paper strips in the trash, wash out the syringes, and you can keep them, and then clean and sanitize your workspace. So that's activity 2. But why don't we go back to activity 1 then for the moment, and we'll finish up activity 1. So if we go back and we look, we're moving pretty slowly here, but we're moving, right? And I don't know if you can appreciate, but we're getting a little bit of a streak there of like a yellow to green, right? This is just moving very slowly. They say 10 to 15 minutes. This is taking much longer. But pretty much what we're going to do when that's done is we're going to remove the chromatography paper strip from the tube. We're going to remove the pin. We're going to place the strip on a paper towel and mark the solvent front with a pencil before the solvent evaporates. So pretty much we want to mark the furthest line that the solvent reached or the isopropyl alcohol reached. So as far as it got up, that's where we're going to mark it. And also take a photo of your results if you would like to at this time, because that's when it's going to be the best. You're going to outline each of the colored spots, so strips, lines, dots, that contain the plant pigments using a pencil and number them from bottom to top. Then you're going to measure the distance in millimeters that the solvent front traveled from the origin line to the solvent front, right? And then, so that'll be the farthest point that the liquid traveled to your origin line. What's the distance? And you're going to record that in data table number 1. And then you're going to measure the distance in millimeters that each color traveled. So you're going to measure from the origin line to the middle of the spot, and you're going to record that in data table number 1 as well. So many factors can affect the rate at which solvents and pigments move through the chromatography paper. So in order to compare the results across the different runs, you can use a ratio called a retardation factor, or RF. And the RF is the ratio of distance traveled by the center of the pigment spot to the distance traveled by the solvent front. All right, so since we're wrapping this up, what I would do then is I would take this out. So let's say this is done. Okay, I would take this out. I would remove the pin. I would lay it on a paper towel. And I'm going to mark the furthest line. And then I'm going to mark the other lines that I see. So in my case, I see the furthest line is here, and I have a little yellow line right here. Okay. So then I would take my ruler, and again, using the centimeter side, I'm going to measure the solvent front. So from the origin line to the furthest point traveled. And for me, that is 4.5 centimeters. And then I'm going to measure the other dots. So I have the furthest line of 4.5, and then the other one I have at 3.8 centimeters. And so in the leaf I used, that's what I saw. I just saw the one dot and then the solvent line itself. All right, and I would record them in data table number 1 and proceed to move through the calculations of the first data table. But yeah, guys, so that's the experiment. Make sure that you're setting up your light and dark. I would suggest actually as a time saver is when you go through activity 2, your prep, your prep's the same no matter what. But when you're doing the light experiment, I would cut out 80, right? And just have all of that prepped, have them sitting, have everything prepped and ready to go. So that when your light experiment is done, you can do the dark experiment. Why they didn't give you 8 syringes is beyond me. So that you don't have to do this over the span of 2 hours instead of an hour. But here we are. But yeah, so if you guys have questions, let me know. I look forward to looking through your labs for this. All right, have a great one, everyone.